you know, I think 4 of those were chosen for presentation by the members of the SMAB, and we're going to have the first of 2 of those now. So first, Nick Ikon from WashU School of Medicine is going to talk to us about the potential of cardiolipin nanodisks as a new... Thank you.
Thank you, Colin. Actually, I'm from UC Berkeley, but close enough. So we heard about several potential therapies throughout this conference. We heard about gene therapy, which seeks to deliver a functional tafazin gene where it's missing. We also heard about enzyme replacement therapy that seeks to deliver the tafazin itself without concern for a functional gene. We're essentially taking this idea one step further in that the main function of tafazin is to remodel cardiolipin into its final tetralinoleal form. So we're looking to deliver tetralinoleal cardiolipin directly and forget about tafazin. And the way that we're seeking to do this is through cardiolipin nanodisks. So what is a nanodisk? It's essentially a type of recombinant HDL particle. So if you take any of the class of small apolipoproteins that are normally tasked with transporting cholesterol throughout the bloodstream, you can purify it and then incubate these apolipoproteins with a dispersion of phospholipids, which will result in these small discoidal structures that are very similar in structure to nascent HDL. Once you have this sort of structure, you can dot this membrane with all sorts of hydrophobic substances. So things like amphotericin B or vitamin A or even small transmembrane proteins. Barth syndrome is kind of interesting in that there is no drug that we're trying to put in there. It's the phospholipid itself that we're concerned about. And indeed, we've tried several different concentrations, and we found that it's possible to make a nanodisk composed of ApoA1 and 100% tetralinoleal cardiolipin. And when we try to form these structures, we indeed see these small disc-shaped particles about 20 nanometers across. So we have cardiolipin nanodisks. And when we treat cells with these cardiolipin nanodisks, in this case we used HL60 cells, which form the Makarian model of neutropenia that I'll talk about a little bit later, we see a huge increase in tetralinoleal cardiolipin, 30-fold increase after just one treatment. And not only does this cardiolipin get into the cells, but within the cells you can follow its itinerary and see that it forms sort of a punctate pattern within the cell. If you stain for mitochondria with something like MitoTracker Orange, you see that there's a very high degree of overlap between where the cardiolipin is and the mitochondria. It's not perfect. You still get a bit of cardiolipin that's going into the cell membrane and such, but for the most part, it's going where we want it to. So it's getting into the mitochondria. Does this make a difference? So for this, we used the model of neutropenia, wherein these HL60 cells, they're promyelocytes. So these are the cells that eventually become neutrophils themselves. And in this model, when you knock down tafazin in these cells, you see a massive increase in apoptosis. When you treat those TAZ knockdown cells with our cardiolipin nanodisks, however, that apoptosis is completely ameliorated. So cardiolipin nanodisks, at least in a cell culture model, have the potential to ameliorate the TAZ deficiency phenotype. But as we all know, cells are one thing, mice are a whole different animal. So we undertook a series of experiments to see how well this therapy could work in a mouse model of Barth syndrome. We started off by just looking at basic tolerability. And it turns out that you can give actually fairly massive doses of cardiolipin to these mice, up to 90 mg per kg, and not see any overt increase in kidney dysfunction, no increase in liver enzymes. At around 150 mg per kg, you do see some elevation, but at this point, you're giving them so much substance that you are doubling the amount of ApoA1 in their bloodstream, so this isn't entirely unexpected. So the other thing is, when you give them a dose of cardiolipin nanodisks, the treatment gets cleared from the plasma fairly rapidly. We've been giving these as intraperitoneal injections due to the frequency with which we would have to administer the treatment. And after an IP injection, around 2 hours, you see peak cardiolipin levels in the blood. But by 24 hours, this is almost back down to baseline. And after 48 and 72, you can barely see anything at all. So it's well tolerated, and it's rapidly cleared. From here, we undertook a longer study using the TAS knockdown mouse model. We used the standard 625 dose of doxycycline. And over the course of 10 weeks, which is long enough to develop the cardiolipin deficiencies, but not enough to see any deficiency in heart function, you can see that despite treatment, all of the TAS knockdown mice remain substantially lower in weight than their wild-type litter mates, whether we treat them every day, every week, or not at all. But we don't see any significant decrease in weight, so further proof that this treatment is probably non-toxic. Unfortunately, the data that everyone really wants to see is after those 10 weeks, do you get an increase in tetralinoleal cardiolipin in the heart and in the skeletal muscle? And unfortunately, that's the data that I can't give you because this last time point was last Friday, and we're still waiting on our collaborators to get that data back to us. But the hope is that next conference I'll be able to get up in front of you and tell you that it was a great success, that tetralinoleal cardiolipin went up, and by then we should have the results of a longer study looking at cardiac function, looking at exercise tolerance, and all of those endpoints. So with that, I'd like to give a thank you to the entire Ryan Lab, especially Jenny Shearer and Betty Su, who were involved in a lot of the preliminary work, to Fengfu Su, who is from WashU, who helped us with a lot of the cardiolipin measurements, and to the NIH for funding our broader nanodisk program, and the Barth Syndrome Foundation for funding specifically the cardiolipin nanodisk set of studies. And I also want to give a shout out to the Brana family, who organizes fundraisers for the Barth Syndrome Foundation in our Oakland community. They've raised a lot of money for the foundation, and just as importantly, they helped researchers like us put a face to the cause that we're fighting for. And it's a pretty cute face at that. So with that, I'll take any questions.
Thanks, Nick, for such a lucid presentation.
Very interesting approach. So there have been some studies suggesting that you are able to transfer lipids from a nanoscale bilayer like this, a nanodisk, into another lamellar bilayer. So even though it seems like a thermodynamic feat to do so, it seems that it is possible. So in principle, yes, that could be possible. But once you've delivered it to the cell, I'm wondering how it's to be targeted first across the outer membrane while it's still within this MSP scaffold, and then delivered into the inner membrane. So that seems like a mechanistically complicated process. Do you have any thoughts on that?
No, that's a great question, but we have absolutely no idea. In fact, this was purely exploratory when we started. We weren't sure if they were going to get across the membrane at all, let alone get into the mitochondria, let alone into the inner membrane of the mitochondria. But while I can, theorize about the potential mechanisms for how this occurs, we haven't looked into it yet. For all we know, this is just some artifact of the way that we're doing things. If it turns out that it works, if it's a feasible treatment, we're going to look more into the mechanism behind it.
Hi, nice study. I just have a question. Are you interested in, or are you concerned about anti-cardiolipin antibodies?
That is a concern, and part of this study, this shorter study, is we're going to look for the development of anti-cardiolipin antibodies in these mice, because that could be a potential downside to this treatment. But it could also not, because the cardiolipin is fairly well packaged up within the nanodiscs, as opposed to, from what I understand, antiphospholipid antibodies usually arise from a strange presentation of the phospholipids.
Nicola, can I ask you, where is the BODIPI label located on the cardiolipin molecule that you're using?
It's attached to the head group.
Okay, so then the question becomes, what portion of the molecule is actually ending up in the mitochondria? Because the classic work of Richard Pagano many years ago showed that when you add lipids to the outside of a cell, you often get hydrolysis and they're reassembled back into the parent lipid in the organelle. Phosphatidic acid is a great example. You add it to the external side of a cell and it'll be hydrolyzed to diacylglycerol on the plasma membrane. That makes its way to the mitochondria and it's reassembled into phosphatidic acid there in the mitochondria. So I guess the question is this an endocytotic type of uptake mechanism which might be delivering to a membrane fraction, or is it in fact a hydrolysis of the parent molecule, and in the fluorescence data that you're showing, it's simply just an assembly of the that glycerol type moiety back into the mitochondria? Because from the fluorescence data that you showed, it looks like it was very universally distributed throughout the cell in addition to the mitochondria. So it might be a more of a general delivery. I'm not saying that wouldn't work, but the point is it may not be truly cardiolipin that's there, but another entity as well. So.
That's a very good point. We could see loss of the BODIPI and have it go off on its own, incorporate into other phospholipids. I guess all I have to say to that is the proof is in the pudding, in that we do see the effect that we would expect to see if cardiolipin had been delivered to the mitochondria. So it's entirely possible that maybe even a large portion of it is broken down, but as long as a substantial enough chunk of it goes to the mitochondria, and I think with our images we see that it does predominantly go to the mitochondria, we would see these effects, and that is indeed what we see.
I guess you could address that with the TAS knockout cells. So my question was just whether these nanodisks can transport other cargoes such as RNAs or proteins into cells and specifically into myocytes.
They can. As far as how it would work within a whole animal, it's hard to say at this point. We're still just coming out of the cell culture studies, but it is possible to put proteins, especially smaller transmembrane proteins, within these nanodisks. And there has been some work looking at using charged phospholipids to attach small RNA. So there are all sorts of possibilities. What we wanted to look at, though, is just to sort of try the simplest approach, just try the phospholipid itself without worrying about anything larger like proteins, because some of these issues were brought up at yesterday's talk with the tafazin replacement therapy. You think that it's hard to push a phospholipid across a membrane, try a whole protein. But you're right, that's very interesting potential experiment.
I think this is very exciting. Just in reply to the question about if it makes it to the mitochondria, how's it going to get across both membranes? But actually, we should think about the mitochondria as a very dynamic organelle. It's not just that it's made and it's static. And in addition to which, if it makes it to the outer membrane of the mitochondria, there are also contact sites between the outer and inner membrane. So I don't think it would be so far-fetched that if it makes it to the outer membrane, it could also get to the inner.
Mm-hmm.
I just wanted to make a couple of very brief comments. So HL60 cells are essentially neutrophils, and they phagocytose. And whatever you give them, any particle, they will grab it and eat it. So to what extent is it can be spread to any other non-phagocytosing cell? That's one point I think, suggest you think. And the other one is that you compensate for potential deficiency of cardiolipin, but you do not eliminate monolysis cardiolipin. And that's what I think you should think about too.
Thank you.
So I have a comment. So we've been studying lipid traffic of phospholipid. So if you culture your cell with labeled phospholipid, and then you monitor where they goes, almost 100% of them go to lysosome. They don't get into mitochondria. I'm not talking about your nanodisk, but if you culture your cell, radio-labeled phospholipid, they're being treated as, just a lipid and being used by autophagy to provide fuel.
Well, I think this one's going to provide plenty of conversation late into tonight, probably. So thank you very much for coming along and telling us about it.